Thank you for joining me today. I just wanted to go ahead and go over the introduction of what I will be presenting to you today. I've chosen the topic of the principles and impacts of recombinant DNA technology. So this is what we will cover today, and I want us to really explore what is recombinant DNA and why is it important? How can this technology be used and what are the potential applications? What are the issues and objections surrounding recombinant DNA technology? The definition of recombinant DNA itself is fairly straightforward. It is an amalgamation of DNA, which is a chimera of sorts, you could think, created through the process of inserting a foreign sequence into the DNA of another species. Seen here is a very simplified diagram of the steps involved. A vector is selected, which is usually a bacterial plasmid, as is the foreign DNA. Then a restriction enzyme is used to cut both strands of DNA at the desired location, as is the plasmid. Ligases are used to successfully join the segment of DNA and the plasmid. Once this is inserted into the organism, which is E. coli shown here in the presentation, the cells undergo transformation to incorporate this new DNA into the rest of its genome. Once the cells divide and proliferate, this new DNA is now a permanent part of its genome. So a more in-depth review of the typical protocol for creating recombinant DNA. The steps would include DNA isolation, which is done through several simple steps itself. The cell membrane must be broken open, which can be done using a detergent or force, like with sound waves. Then proteins within the cell itself are going to be degraded. This is done to ensure that they do not precipitate with the DNA in the next step. That degradation would be done using a protease enzyme. Once this is completed, the DNA can be precipitated or made solid with the addition of cold alcohol. Once it's precipitated, the DNA can be isolated from this point with the use of a centrifuge. DNA amplification is then completed using polymerase chain reaction, a step we've used in the lab several times, usually referred to as PCR. Detecting the DNA at that point can be done through a few different methods. You can do that through utilizing UV absorption, because we are aware that DNA absorbs UV light at 260 nanometers. You can use stains or dyes, and gel electrophoresis is actually a very common method of detecting the DNA. Vectors. So after your DNA amplification, the selected sequence is then inserted into a vector, which can range from a plasmid to an artificial chromosome. There are several benefits to using a vector. They can aid in streamlining the selection process because you have a marker or an identifying gene within the plasmid to highlight which organisms have the desired gene. For example, the plasmid not only has the newly inserted gene, but also has a gene for antibiotic resistance within it. Growing this new transgenic bacteria in an antibiotic-enriched medium would ensure that only populations containing your vector would thrive. An additional bonus with the use of vectors is that they can be stored outside of the organism within a freezer for future use. So how is the DNA placed into a vector? Restriction endonucleases are enzymes used to cut DNA at specific loci. The cuts can be blunt, which means they line up and are cut straight across the DNA, or sticky, with the backbones cut at different points, resulting in small overhanging sections of single-stranded DNA. After they're cut, the pieces of DNA can be mixed together and linked with the addition of another enzyme, DNA ligase. Again, I referred to that on the previous slide. Just for future reference, sticky ends are actually preferred and typically yield better results in the lab. So once all of this has been completed, you want to read or sequence the DNA, and that can be done with a variety of methods. Older methods, such as the Sanger method, which is very similar to polymerase chain reaction, can be used to create clone libraries to shotgun entire genomes. This was actually the main method utilized within the Human Genome Project, which, as we are aware, is a massive genome. So you can really use this with a variety of organisms. Newer methods, dubbed next generation methods, include 454, Celexa, or Illumina sequencing, and are more computer program or algorithm based sequencing. And the most current methods are nicknamed the third generation, see the theme going here, include ion torrent and nanopore sequencing. So what are the applications we'll see with recombinant DNA? Transgenic DNA technologies opened many new doors within the medical field. Researchers can now attempt to create multivalent vaccines, producing cross-protective immunizations would be beneficial for a number of reasons. For example, there was a case study done for a leptospirosis multivalent vaccine. Leptospirosis, as we know, is an endemic disease in many tropical regions around the world. And in this instance, Garba and his colleagues have been working on a multi-epitope DNA vaccine, which has shown some promising results. It was shown that the chimeric DNA immunization stimulated both agglutinating and neutralizing antibodies. These antibodies reacted against multiple pathogenic serovars and reduced leptospiral colonization within the kidneys or renal system. Other amazing feats include the production of anti-cancer medications using recombinant DNA technologies. Most of the therapeutics being created currently focus on tumor death and stimulation of the immune system in response to cancer cells, which is definitely the future of cancer medicine, I believe. 50 years from now, we'll look back at chemotherapy and radiation with what the heck were we even thinking here? A more recent development of recombinant DNA technology is the gene therapy tool dubbed CRISPR, the apt nickname standing for Clustered Regularly Interspaced Short Palindromic Repeats, which is a mouthful, heck, CRISPR. So, it's a novel approach to gene editing, and this tool has proven to be more precise and efficient than previous technologies, allowing researchers to deftly correct disease-causing mutations within the human genome. CRISPR was created from the adaptive immune system in prokaryotes and is partnered with bacterial endonucleases, commonly referred to as Cas9, to complete this cut, remove, add sequence, which we've discussed several times within this presentation. This tool has had great success in treating debilitating diseases such as cystic fibrosis, hereditary tyrosinemia, and muscular dystrophy. Moving on, we see that these technologies can be utilized to create genetically modified fruits, vegetables, animals, microbes, and even biopolymers. Such research can be carried out to modify plants to yield better nutritional values, such as golden rice, which is high in vitamin A. This nutrient is normally missing from rice, which is a food in many cultures. It's a big staple worldwide. The absence of this vitamin can lead to abnormal neurological developments and an increased risk of blindness in childhood. In addition to changes with nutritional content, recombinant DNA technologies can be used to modify foods so that they are more resistant to microbials, such as fungi and bacteria. Lysozymes are an effective way to reduce pathogenic bacteria presence within the food industry, as they prevent the colonization of microbial organisms. Researchers are currently working on methods of engineering a lysozyme-infused or embedded coating to apply to foods as a means to increase their shelf stability. Another excellent example of the power of recombinant DNA is the recent work in creating biopolymers. Taking the lead from nature, researchers have attempted to recreate the production of natural fibers, such as spider silk, in synthetic ways. This natural polymer is unique in the sense that pound for pound is actually stronger than steel or Kevlar, yet it's flexible. There are many challenges with mass producing these fibers in a lab setting, as spiders cannot be farmed due to their aggressive or territorial behaviors. Therefore, a biotechnical approach using recombinant DNA is the best solution to mass-produce spider fibers. In recent years, researchers have been able to engineer bacteria, yeast, insects, plants, mammalian cell lines, and transgenic animals, to name a few, to express silks. A key aspect to using recombinant technology means that scientists can manipulate the genome in many different ways, allowing for customization and a variety of products. As society becomes increasingly aware of the impacts of fuel processing, a keen interest has been developed in finding a more ecologically sound method of fuel production. Thinking of the future of our planet, researchers are turning to recombinant DNA technology in the hopes of creating biofuels. Most of the research currently being carried out is focused on microbes such as yeast and E. coli, as a plant-based biofuel production is more limited due to the space required and the short lifespan of the crops. There are several ways to manipulate E. coli to create biofuels from alternative carbon sources, such as cellobionic acid or glycerol, to produce the respective isobutanol and bioethanol. Arizona State University, my alma mater, has actually developed the Center for Bioenergy and Photosynthesis in order to investigate cleaner fuel production. Specifically, they're focusing on hydrogen, ethanol, and biodiesel from photosynthetic organisms. Current research projects explore the metabolic manipulation of organisms for bioenergy production, as well as developing photosynthetic processes in the hopes of one day creating artificial photosynthesis in non-biological systems. These projects are centered around the foundations of recombinant DNA technologies and bring together some of the brightest minds today in sustainability. There have been concerns voiced about recombinant DNA technologies and their use. Some are concerned about the potential that gene editing could be taken too far, as seen with Dr. He Jiankou's experiments in 2018. His work with twins Lulu and Nana were clouded with controversy, as he claims to have created HIV-resistant babies using CRISPR. Did he go too far? Many think that he did, raising concerns about the practices and ethical accountability surrounding this case. Recombinant DNA and gene editing technologies can literally save lives, but where do we draw the line? Even though scientists can create designer babies, should they? In 1974, the United States founded the NIH Recombinant DNA Advisory Committee, referred to as the RAC. This committee was created to aid in advising the National Institutes of Health about recombinant DNA practices. Their main goal is to ensure that research is being completed in a safe and ethical manner. The RAC also plays a key role in keeping politics at bay, as much as possible at least. While the Human Genome Project was in full swing, the U.S. Congress was actually interested in human genetic engineering and proposing legislation to regulate recombinant DNA research. Thankfully, the RAC's history of ethically based guidance was able to squelch these statutes, and none were passed. To keep it short and sweet, I just want to quickly review why I chose to present this topic for you. Recombinant DNA technology has come a long way since its inception in the 1960s. Scientists are still discovering new ways to utilize this technology, with uses ranging from the medical field to agriculture and bioenergy. Overall, it has been shown that the benefits and value this technology provides far outweighs the potential ethical and legal challenges that may arise. If you wish to learn more about the current research on recombinant DNA technologies, these are a few excellent sites that you can visit. And these are the references for the information that I had found to present to you today. Thank you.